Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Chance Millett-Bangs from the Business Review, and I will be your host. It is our pleasure to have Bio-Rad with us today, who will be discussing a new way to detect cancer mutations in cell-free DNA using Droplet Digital Polymerase Chain Reaction, or ddPCR. ddPCR is an approach to provide high-precision absolute quantification of nucleic acid target sequences. Today's guest speaker is Dr. Dawn Shelton, Senior Application Scientist at the Digital Biology Center. I'd like to welcome you to our webinar platform On24. This webinar is browser-based, so if you disconnect, please click the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the Q&A widget at the bottom of your screen, or you can use the questions box at the top left-hand corner of your screen. We will allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. We also have a few extra resources in the green resource list widget for you to view or download at your leisure. Please use the help widget if you require any more assistance. So now, a little bit more about our speaker, Dr. Shelton. Dr. Shelton received her PhD in Oncological Sciences from the University of Utah Huntsman Cancer Institute. She has worked both in academia and industry on a variety of cancer types, such as colon, breast, uterine cancer, and leukemia. She is now a senior scientist at the Digital Biology Center, focusing on developing applications and collaborations for ddPCR in the field of cancer. So, without further ado, let's get things started. Dawn, over to you.
Thank you, Chance. Hello, my name is Dawn Shelton, and as Chance mentioned, I am a Senior Applications Scientist from the Digital Biology Center in Pleasanton, California. I was hired to help translate our new Droplet Digital PCR technology into applications in the cancer field. I'm here today to talk to you about ddPCR and the liquid biopsy. Droplet Digital PCR is the third generation in the evolution of PCR, moving from qualitative to absolute quantitative measurement. In real-time PCR, the effects of inhibition, either by primer efficiencies, quality of sample, or chemical inhibitors, are always a concern. Any cause for delays in the CQ values alters fundamentally the accurate measurement of the template, leading to greater errors. With Droplet Digital PCR, each droplet is thermocycled to endpoint individually and read as either a positive or a negative for the target of interest, hence the digital aspect of ddPCR. There are no standard curves, and ddPCR is much more tolerant to PCR inhibitors. ddPCR enables highly precise and sensitive nucleic acid quantification in a convenient high-throughput workflow. We take a typical 20 microliter reaction, partition the reaction into about 20,000 droplets, run the plate in a standard 96-well thermocycler, and then read each droplet in a flow cytometer-like fashion. Droplets containing the target become fluorescent and are counted positive by the droplet reader. Droplets without target are counted negative. One reaction effectively goes from analog to thousands of discrete yes-no answers. The key technical advantages of ddPCR are summarized here. First, because each droplet is a yes-no answer for your target, ddPCR simply counts the number of input molecules. Your output in copies per microliter tells you how many amplifiable DNA molecules you screened. There is no standard curve, and because we read at endpoint, there is minimal worry about PCR efficiency. This is especially important on degraded clinical samples. Second, a digital readout of 20,000 gives a high precision and reproducibility, allowing us the ability to discriminate small differences in nucleic acid content. This high precision leads to high sensitivity, which enables detection of rare events like cancer mutations or virus in plasma. In effect, through sample partitioning, droplets serve to titrate out high copy backgrounds, effectively increasing the signal-to-noise ratio of a rare target. In this example, the bulk sample has a mutant species present at 40 copies and 40,000 copies of wild-type molecules. When the sample is partitioned into 20,000 droplets, 40 droplets will contain a single mutant target, now at a ratio of 1 to 3 mutant to wild-type background. This year we have brought new products to market to meet the needs of our growing customer base, the cancer researchers. We've recently released the AutoDG, a machine that automates droplet generation up to 96 samples at a time. There is minimal hands-on time, a high throughput format, and consistent expert quality droplets from any user. We've also been expanding our catalog of wet lab validated prime PCR, mutation detection, and copy number variation assays, optimized for the ddPCR platform. We now have assays that target the top mutations and genes present in Cosmic, with thousands of expertly designed in silico assays for the less prevalent targets available today. This is an example of one of our validation data set from one of our most popular mutation detection assays for BRAF V600E. For our mutation detection assays, we use a single primer pair to amplify the mutation site, then two competitive probes to detect mutant or wild-type copies. Mutant is in the FAM, wild-type is in the hex. Shown here on the left is an example of a wild-type only control well and a mutant-positive control well on the right at 0.1%. The 2D plots shown here display each droplet in the reaction. The no template droplets are in gray, the wild-type single positives are in green, and the mutant positive droplets are in blue, with the double positive mutant and wild types shown in brown. The limit of detection plot down below shows 0.1% mutant detection in a single well, which is 1 in 1,000, with clear separation from the wild-type only negative control as indicated by the dashed line below. This is the same assay with a 10-fold lower LOD in 4 merged wells. Each of our validated assays are tested to 0.1% in a single well, but all can achieve much greater sensitivity by the addition of more wells, more target DNA, and the merging of data. In addition, one of the main strengths of ddPCR is the precision counting of input templates, which means 3 users, 3 instruments, in 2 different locations produced near identical results from 1% to 0.01% mutant fractional abundance, as shown here. On the right-hand side, you can see the absolute concentration counts with the 3 users as well as the 3 machines nearly overlapping, and on the left, the percentage mutant. Chance, on to you.
So now it's time for our first poll question that we'll be having in today's session. So if you can please pick the answer that is relevant to you and then press submit. So our first question that we'll be having is, what sample type do you most commonly use? Your selection of answers are formalin fixed paraffin embedded or FFPE, frozen tissue, plasma, serum, whole blood, or finally other. So Dr. Shelton, just while the audience are voting, can you please tell us why you decided to ask this question in our session?
The large number of people using ddPCR right now appear to be cancer researchers, and the variety of different kinds of sample types are of interest to us for the quality of DNA that can be expected, the inhibitors that might be present, and the variety of samples that people are using.
Thank you very much. I now believe that's enough time for the audience to have voted, so let's quickly move on to our results. Okay, so plasma seems to be our front runner there, but it seems to be pretty even across the board other than that. So Dawn, would you like to discuss these and then carry on with your presentation?
Thank you, Chance. Yes, plasma seems to be the forerunner here, which is good since it's a liquid biopsy talk, which brings us nicely to our next slide. The liquid biopsy is the holy grail in diagnostics since it is largely non-invasive and believed to be more representative of the overall body rather than a biopsy of a tumor in a single location. In addition, multiple biopsies over time are considered impractical, expensive, and can be downright dangerous for the patient. However, there is very little DNA in plasma, about 5 to 50 nanograms per mil in advanced cancers, and the quality of the DNA is very poor, mostly apoptotic fragments. Most of the DNA is also wild type in nature, arising from the non-tumorigenic cells. These limitations have prevented the use of plasma DNA as a diagnostic test up until very recently. Shown here is data from the lab of Luis Diaz at the Johns Hopkins University. In this figure, you can see the percentage of patient samples on the y-axis that have detectable cell-free DNA in the plasma from different tumor types in advanced disease on the x-axis, indicating that about greater than 80% of the patients with metastatic disease have detectable levels of cell-free DNA. The figure on the right indicates the number of copies detectable per 5 mL of plasma from each of the tumor types. They further go on to show that the levels of mutant cell-free DNA vary by stage of disease and can provide therapeutic, predictive, and prognostic information, making it promising for monitoring residual disease. The use of tumor genotype to guide patient treatment is routine in a variety of cancers, including melanoma, colorectal, and breast cancer. Roughly 50% of the melanomas have activating mutations in BRAF, and V600E in particular accounts for an estimated 80 to 90% of that half. Personalized cancer treatment in melanoma requires detection of activating BRAF mutations. So we formed a collaboration with Dr. David Polsky at the New York University Langone Cancer Center to evaluate the potential of digital PCR to measure cell-free DNA from the plasma of patients undergoing targeted therapy or immune checkpoint blockade in melanoma. They evaluated 115 samples from 25 patients over the treatment course. Initially, several platforms were tested, and ddPCR performed exceptionally at detecting a single mutant copy across the range of DNA input amounts when a sufficient number of replicates were run to overcome stochastic distribution effects. Shown here are 3 DNA dilution series at 300 nanograms, 330 nanograms, and 3 nanograms DNA with 10-fold dilutions ranging from 10% to 0.1%. ddPCR was able to resolve a single copy at 0.01% in this experiment, shown in red at each of the different levels. This is an example of one patient from the study whose mutant BRAF levels tracked with progression of disease. Shown here is their response to BRAF and MEK inhibitor treatment, stable disease, SD, for a short time, and rising mutant DNA levels with disease progression. For this patient, rising cell-free DNA levels were detected 43 days prior to standard clinical detection, with a steady average of 84 days prior to scans. In 65 out of 71, or about 92% of the plasma samples, changes in the DNA levels trended with the disease status as evidenced by radiographic scans. Though much work still needs to be done, this study and others like it show promise for monitoring residual disease and recurrence using ddPCR. EGFR signaling is a significant driver of metastatic colorectal cancer, and blocking EGFR signaling is one of the major treatment options. However, downstream activating mutations in KRAS block the benefits of anti-EGFR therapy in colorectal cancer in about 40% of the CRC cases, and are therefore counterindicated for anti-EGFR therapy. There are 7 mutations present at greater than 1% population frequency, and an additional 15 more present at less than 1% frequency in COSMIC. To address the need for screening a greater number of mutations with a very limited biological sample, we developed the KRAS screening multiplex. This assay is designed to screen for the 7 most common KRAS activating mutations in colorectal cancer samples. It was 1 set of primers, 1 wild-type probe in the hex, and 7 mutant probes, each in FAM, covering the most common G12, G13 mutations. The 2D plot shows an example of a positive control of this screening assay being used on a 7-mutation mixed positive control DNA sample. This assay is merely a screening assay, so it is a mutation yes or a mutation no. This concentration plot, shown with G12D, represents the validation data for the KRAS multiplex assay. The KRAS mutant DNA, indicated in blue, was run in a threefold dilution series in a constant background of about 40 nanograms human wild-type genomic DNA in green. This established sensitive single-well detection of KRAS mutations down to 0.2%. The plot on the right shows how sensitivity is a function of the percent mutant and the number of amplifiable copies screened. This is an ideal world example and to be used as a reference point since it assumes perfect DNA sample, perfect assay, and no false positives. Chance, the next poll question.
Thank you very much. So it's now time for our second poll question that we'll be running. With this poll question, you can select multiple options, so please select all that apply here. So this next question is, what sample extraction methods do you currently use? So your options are home brew, DNeasy blood and tissue kit, blood and cell culture DNA kit, QIA-AMP circulating nucleic acid kit, QIA-AMP virus spin kit, trizol or phenylchloroform, Maxwell DNA purification kit, or finally other. So Dawn, can you just tell us what kind of results you are expecting from this poll question?
Hi, Chance. Thanks. Typically, we see our customers using the QIAGEN kits and as well as the Maxwell system from Promega. But depending on your region, different methods are predominant, so we're interested in what our customers are using. Each particular kit comes with its own limitations and benefits, pros and cons, and so we're always following up at this point since ddPCR measures quality quite well. We're measuring right now the variety of different sample types and how that impacts it.
Thank you very much, Dawn. So now that we've given the audience a little bit of time to vote, let's quickly move on to our results. Okay, so it's pretty even across the board there. Dawn, would you like to briefly discuss this before we move back to our presentation?
Yes, thanks, Chance. So the CNA kit, the QIAGEN circulating nucleic acids kit, is the predominant kit that we've seen both in the literature as well with our customer base. So I'm not surprised with this, and it's an excellent kit. So here is an example of what cell-free DNA would look like in Droplet Digital PCR. These are the 2D plots of the KRAS screening multiplex assay on cell line DNA on the left, FFPE in the middle, and cell-free DNA on the far right. As you can see, there's a fundamental difference in the plots representative of the quality of the DNA sample inputted. Cell line DNA gives clear separation with the 4 clusters, while highly damaged FFPE tends to have intermediate fluorescent droplets, which we call RAIN, due to the presence of non-amplifiable DNA and inhibitors, while apoptotic cell-free DNA tends to have an intermediate phenotype. If you're examining FFPE in your own research, this phenotype is perfectly normal. And it is absolutely routine with our experience that oftentimes you get less than 10% amplifiable DNA of the amount of DNA that you put into a reaction for FFPE. These are the concentration plots and a fractional abundance graph, percentage mutant, of wild-type KRAS in green and mutant KRAS in blue, with a percentage mutant in orange from 6 MD Anderson Cancer Center cell-free DNA plasma samples, demonstrating the range of mutant detection in real samples. We take a fairly conservative approach towards calling a sample positive based on non-overlapping 95% confidence interval error bars between the appropriate wild-type control and samples. For instance, in sample number 1, it falls below the dashed line, while sample number 2 falls above. We call sample 1 a negative for the KRAS G12AC, D, RSV, and G13D, and sample 2 an above positive for at least one of these mutations. This is data from Helen Huang's very first experiment showing excellent concordance between CLIA-tested tissue samples and ddPCR from the plasma, both in duplex and multiplex assay formats. The one apparent discrepant sample, number 1, was actually a mutation not present in the screening assay, Q61H. Therefore, it had shown up correctly as wild type for the 7 KRAS G12, G13 mutations screened. For in-house testing of the KRAS screening multiplex, we purchased plasma samples from Conversant Bio, representing 12 patients and 52 draws, with 5 patients testing KRAS positive by CLIA tissue testing at diagnosis. We also purchased normal samples from ProMed-Dx, representing 12 pregnant patient samples. We used the QIAGEN CNA kit to process the samples. In this first graph, cell-free DNA was quantified by using duplexed copy number assays on the 12 normal and 12 cancer patient samples, a total of 64 draws. Using ddPCR, it is clear that the amount of amplifiable cell-free DNA is highly variable, both across groups and across the draws for a given patient. The median value of cell-free DNA from normal samples was about 3.8 nanograms of amplifiable DNA, while the KRAS mutant samples had a median of 27.4 nanograms and KRAS wild type had a median of 18.1. The large error bars seen in the KRAS mutated and KRAS wild type patient samples are due to the variability seen in the longitudinal draws from the same patient. This patient had 15 draws over a 7-month period, and as you can see, the amount of DNA per draw is highly variable. Unfortunately, we don't have any data to correlate with these particular time points based on treatment or response. We then use the KRAS multiplex assay to detect KRAS mutations in normal and patient cell-free DNAs. The plasma samples were taken years post-diagnosis, and as such, we were not surprised to only find 1 KRAS-positive patient sample. This was present at about 1%. None of the normal or KRAS wild-type patients revealed KRAS mutations using this strategy. Further analysis using individual duplexed KRAS mutation assays identified the M3K patient sample as KRAS G13D. Wild-type controls were used with each assay to identify the correct false positive rate for each assay, and the positive calls were made by non-overlapping 95% confidence interval error bars. Since we did not have enough sample left to screen all of the individual mutations, and we had already determined the absolute concentration of the sample, we used the So Advanced Preamp Supermix from Bio-Rad to generate enough sample to identify the individual mutation type. There was some amplification bias, as is often seen with preamp, which altered the 1% mutant to 0.64% mutant after preamp. But since we had an absolute measurement from the initial screen, a qualitative answer was perfectly acceptable. Another advantage of ddPCR is the ability to identify PCR inhibition by the 1D or 2D plots. Shown here is one sample loaded at 6 microliters and 2 microliters, where you can see an obvious suppression in the red, with the red arrow of fluorescence amplitude, representing the PCR efficiency as compared to the controls. As long as there is a clear separation between the droplet clusters, accurate quantification can be made. When separation is poor, the fractional abundance will generally still remain accurate. Despite the obvious inhibition in these samples, the absolute concentration when back calculated is fairly equivalent. To further evaluate the performance of the KRAS screening kit, we also purchased colorectal cancer FFPE samples from Advanced Tissue Services. We used the QIAGEN DNA Easy Kit to process 16 KRAS mutant and 4 grossly normal samples. We first extracted the DNA from the 16 metastatic colorectal cancer samples and the 4 wild-type FFPE samples using standard protocols. We used duplex copy number assays, AP3B1 and E1F2C1, to estimate the fraction of sample that could be PCR amplified. 13 out of 20 of the CRC samples had less than 5% amplifiable material. We found that the amount of amplifiable material is not linearly related to the amount of the DNA detected by nanodrop. qubit is often a better reading, but also still quite inaccurate as a basis of amplifiable DNA. For example, several samples with greater than 100 nanograms of material by nanodrop had less than 10 nanograms amplifiable material, which has significant implications for the interpretation of the data and sensitivities of genomic tests. We have found that the yield of amplifiable DNA from FFPE samples is highly variable and not well assessed by spectrophotometric analysis. Shown here are measurements made of Horizon FFPE reference samples for each of the 7 KRAS mutations measured by specific duplex assay or the KRAS multiplex. Here we demonstrate quantitative measurement of FFPE using both duplex and multiplex assays. As you can see, the concentrations are nearly identical with overlapping 95% confidence intervals. We use the KRAS screening multiplex to detect 7 most common KRAS mutations in the 16 colorectal cancer samples. 6 samples had a high mutation fractional abundance of greater than 3%. We also use the 7 individual KRAS mutation assays to identify the dominant or sole KRAS mutation picked up by the initial screen, shown below in the duplex format. For example, M2K was a 2% mutant, and the single assay shows that it was a G12D. Our data was consistent with COSMIC. The majority of the KRAS mutations, greater than 80% in these colorectal cancer samples, contained either G12D, G12V, G13D, or G12S. Further, similar to the Horizon references, measurement of FFPE samples by duplex and multiplex assays correlated strongly with an R-squared of greater than 0.99. And again, as mentioned previously, you can see that the 1D and 2D plot information can be used to visualize and troubleshoot your experiment for PCR inhibition. For this sample, F7K, 20% of the material is amplifiable, but inhibitors are present at 5 microliter loading, impacting the positive amplitudes on the left. Loading less sample or making a dilution allows better amplification and accurate quantification. Chance, the last poll question is for you.
Thank you very much. So this once again is another poll question that we've come up to, which will be our final poll question. Again, you can pick multiple answers here, so please select all of those that apply to you and then press submit. So our question is, what multiplex mutations do you wish to screen for? So answers are KRAS codons 12 and 13, KRAS codon 61 mutations, PIK3CA exons 9 and 20 mutations, BRAF V600 mutations, NRAS codons 12 and 13, or finally other. So for the final time, Dawn, can you just explain to us why you decided to ask this question in our session?
So we have now released the KRAS multiplex assay for G12 and 13, and we are assessing which, for our new product development process, which are the next highest hitter multiplex assays desired by our customers.
Thank you very much. I believe that's enough time for the audience to have voted. So for the final time, let's move on to our poll results. Okay, so that's pretty even across the board there. So Dr. Shelton, would you like to briefly discuss this before we move on to the final part of your presentation?
Well, it's excellent to see that KRAS 12 and 13 is highly selected. BRAF V600E is the one that I immediately was planning on working on next, so that's reassuring. And further multiplex are to be released in the next year. So switching gears slightly, we're going to move on to copy number variation or alterations in cancer. This is kind of a bit more of a black box field. It's been known for a really long time, but it's been incredibly hard to measure. So another field that's being very quickly adopted by our users with the advent of whole genome data is the study of copy number alterations or variation. Roughly 12% of the genome has been found to be copy number variable in the normal population. These variations may be caused by duplications, deletions, inversions, or translocations. In addition to small regional changes, the entire chromosomes may be deleted or duplicated in disease states. Germline variations might lead to susceptibility in disease or adverse events to certain drugs. In cases post-onset of cancer, the cancer process itself acts as an accelerated genetic selection process. This somatic expansion and selection process results in a survival of the fittest cancer cell race, leading to many somatic copy number alterations and genomic imbalances. The implications of these variations are not currently very well understood. However, one documented example would be the amplification of HER2 in breast cancer. Studying copy number variation in cancer is not a trivial exercise. For ddPCR, we have found it requires short, matched amplicons for your target and reference. When using damaged DNA types like FFPE or cell-free DNA, the smaller the assay footprint, the more fragmented DNA you can detect. Shown here are two assays against MYC, one designed for cell line DNA and the other designed for fragmented samples. When the reference gene and target are not matched, you can get significant alterations in your copy number, in your CNV copy number, in this case a 1 versus 2 copy difference on the right. Tumor heterogeneity also causes problems for copy number variation studies. Identifying the most appropriate reference for your tumor type is critical. We recommend using multiple references and multiplexing. Normal population references, especially TERT, are not appropriate for many cancers as they are frequently deleted or amplified in certain cancer types. To this effect, wrong slide. So due to the heterogeneous nature of tumors, a single reference is often inadequate for calling copy number. Shown here are 4 cancer cell lines tested with HER2 and 2 different references. In the first 2 cell lines, the HER2 copy number is identical for these cell lines. However, for SKNMCs and HS578Ts, the choice of reference alters the copy number call. To address this, we have wet lab validated about 60 common pericentromeric reference assays corresponding to each chromosome. Whatever your cancer type of interest is, you can find a validated reference assay for it. For a blind approach, we asked our bioinformatics group to figure out a method to determine the average genome equivalence of a sample, or a baseline reference number. They determined that with 8 references, at least 4 will be informative 99% of the time. By tossing out the outliers and keeping the middle 50% interquartile range, you can establish a good reference number for your target of interest. Using this method, we tested 6 colorectal cancer cell lines and 4 targets, MYC, EGFR, SMAD4, and TP53. We then compared our results with the COSMIC data. Shown on the right in red is ddPCR, and the COSMIC calls are in blue. And what you can see is the COSMIC and ddPCR calls are almost identical. ddPCR and SNP array data in COSMIC gave high correlation with an R-squared of greater than 0.98. The exceptions were copy number states higher than 10, with the dynamic range of arrays showing suppression. For example, shown on the right are the MYC copy numbers, where COSMIC data suggests a copy number of 14, and ddPCR measures about 10-fold higher. This data and much more are recently released in a paper by Steve McCarroll's lab. So in this recent publication, Jeffrey Oxnard and colleagues at the Dana-Farber Cancer Institute used ddPCR to monitor patient treatment in lung cancer. These researchers want to use cell-free DNA from patient plasma to track and detect EGFR sensitizing and drug-resistant lung cancer mutations such as T790M over the course of treatment. To evaluate this possibility, they performed rare mutation detection of multiple EGFR, KRAS, and BRAF mutations on cell-free DNA from lung and skin cancer patients. Here I highlight the work on EGFR in lung cancer. First, they use conventional assay validation approaches to establish sensitive detection of EGFR, L858R, and T790M mutations, down to less than 1 in 10,000 copies using ddPCR. They then apply these assays to plasma from lung cancer patients undergoing therapies. One example of this profiling is shown, and you are able to see a serial non-invasive mutation detection during treatment, including the rise of mutational load as brain metastases evolve. In another compelling finding, the team was able to show, using ddPCR in 6 out of 9 patients tested, reemergence of the EGFR mutations detectable up to 4 months ahead of objective disease progression using radiographic imaging. This sensitivity has the potential for massive patient benefits, both in terms of comfort and the ability to quickly iterate on treatment options. In this paper, published last February, what is remarkable is that aside from detecting mutations in plasma, this is the first study to examine early-stage disease using ddPCR successfully. All previous studies have focused on late or metastatic disease because there is much more circulating DNA available. Our collaborator at Johns Hopkins was interested in studying plasma markers in early-stage breast cancer patients, focusing on PIK3CA mutations. Typically, patients with early-stage disease have surgery to remove the tumor, then chemotherapy. However, 60 to 70% of these patients are treated with chemotherapy when surgery alone would have cured them. To prevent overtreatment, this group decided to look into whether or not the patients being treated were necessary. 29 patients were enrolled in this prospective study. The clinicians took both preoperative blood samples and postoperative blood samples, in addition to the FFPE samples of the excised tumors. The tumors were sequenced by Sanger sequencing and you can see the results on the right, and 10 patients were found to have PIK3CA mutations. However, when digital PCR was applied to those exact same samples, an additional 5 patients were found to have PIK3CA mutations. This was due to the greater sensitivity of ddPCR. When digital PCR was applied to the plasma samples, as shown on the left, 14 samples were found to be mutant, closely paralleling the FFPE data. Several weeks after surgery, 5 patients continued to show mutant DNA in their plasma, while 12 did not. This meant that in this small prospective study, that ddPCR had 93.3% sensitivity and 100% specificity. Since our first publication of ddPCR in 2011, our customers and collaborators have found hundreds of ways to use Droplet Digital PCR in their research. This speaks to the ease of use and the affordability of the system, as well as the technical advantages of ddPCR. We have now over 201 publications to date, and about a quarter of those are cancer-related. Our applications group at the Digital Biology Center, as well as our customers, are putting ddPCR to use in multiple areas of cancer research, which includes low-frequency mutation detection in clinical or degraded samples. Sanger sequencing is sensitive down to about 20%. Next-gen sequencing may be 5% or 1% at best. And ddPCR gets down to less than 0.1% in even a single well. A lot of work is being done with, as I mentioned before, with copy number alterations and comparing that to arrays. Also, some very interesting work being done is using ddPCR to measure TILs, or tumor-infiltrating leukocytes, and circulating tumor cells, CTCs. Additional applications, areas of research, are microRNA detection, methylation quantification, and telomerase or enzyme activity assays like telomerase telomere repeat amplification protocol. For more information, I encourage you to watch our previous webinar on ddPCR cancer applications, as well as our webinars on specifically CNV, copy number variation, gene expression, multiplexing, and tips and tricks. So thank you for your attention, and I'd like to open the talk for questions.
Thanks, Dr. Shelton. So as was just mentioned, it's now time to move to our Q&A session. If you haven't submitted your questions so far, then please do so by clicking on the Q&A widget at the bottom of the screen or by using the questions box. As I go through these questions as well, different ddPCR cancer publications will be displayed on screen. To view this full list, then it is uploaded into the green resource list widget. So our first question is, can ddPCR be used for DNA methylation discrimination, especially in cell-free DNA?
Absolutely. So last year I submitted a poster to, I believe it was AACR. We were testing whether or not we could detect cell-free DNA, methylated cell-free DNA. The problem with the sample type is, again, that cell-free DNA has very, very low amounts of DNA, and then when you do your bisulfite conversion, you get even a lower percentage of DNA recovered due to the conversion process being fairly harsh. But it is possible and absolutely measurable.
Thank you very much. So moving on, our next question is, have you tried to use the reply G from Qiagen to amplify the sample first? Did you see any difference from no amplified?
I'm actually not familiar with reply G from Qiagen. We have not used that kit, I assume, and I will certainly make a note of that and look into it.
Okay, thank you very much. Our next question is, if I understand it correctly, the assay is only applicable to detect known mutations. Is this correct?
Like other PCR-based methods, digital PCR is based on knowing what your sequence is and what your mutation is. That being said, there are creative assay formats where, for instance, the Oxnard paper performed one where in a deletion assay, we call it a drop-off assay, where you have a probe that is directed towards a specific wild-type sequence, and if some unknown mutation or some unknown deletion under that site occurs, then the probe will drop off, and there is, based on the quantification, you can tell there is a mutation there, and you don't necessarily know which one it would be.
Thank you very much. So if I now move on, our next question is, can we sequence the amplified product from ddPCR output?
Yes, actually several of our customers are already doing that. It was a rather surprising thing after the release of the first machine that people wanted to do that so quickly. The droplets can be lysed, essentially, and the PCR products purified out and dropped easily into sequencing.
Thank you. Moving on, our next question is, how is this relevant to prostate cancer? What can we use?
How is this relevant to prostate cancer? I mean, you can certainly use it for any kind of tissue biopsies. I'm not exactly sure what the levels of cell-free DNA are in prostate cancer. We'd have to look back at the Luis Diaz paper and to identify, but I do believe I've seen papers looking at prostate cancer.
Thank you very much. Just before I move on to our next question, we have had quite a few requests in for a copy of the presentation slides. Just want to make the audience aware that a copy of the slides as a PDF will be available for you to download from the green resource list widget in the on-demand version of this webinar. So if we move back to our questions now, our next one is, can you achieve sensitivity higher than 1 out of 20,000? So that's a sensitivity higher than 1 out of 20,000.
Oh, absolutely. So the 1 out of 20,000 comes up frequently because of, in one well, we have 20,000 droplets, but we have this beautiful ability to merge our wells into what we call meta wells. So you can, in one plate, in theory, you could merge all 96 wells and have, 20,000 times 96 droplets. So you can get much, much higher sensitivity, and the truth is, by that level, the main thing limiting you is generally the amount of sample you have. To do, for instance, plasma cell-free DNA to 1 in a million, in most cases you have to exsanguinate a person. So it's generally the first limiting factor is sample input, and the second limiting factor might be your assay design.
Thank you very much. So our next question is, so you need DNA, and sort of related to this, what is the minimal amount of DNA for ddPCR reactions?
Good question. So minimal amount of DNA, we can detect single copies. The question really becomes a game of numbers. How much are you going to trust if you see one single positive mutant? I mean, what's your confidence in calling that a true positive? So that becomes a game of knowing how many negative controls that you had to kind of measure what your confidence level in that single positive would be. But as it's 3.3 picograms of genomic DNA is typically one haploid genome, you can detect just one haploid genome.
Thank you very much. So our next question is, what is or how is ddPCR any different than any other PCR machine? What's unique about it?
The main things that ddPCR can do is in qPCR and the old-fashioned first-generation PCR, we were always guessing at how much of the initial template we were measuring. Maybe guess is not a great term, but you could back calculate to how much you thought, based on the dynamics of amplification, how much your starting material was. And truthfully, with digital PCR, since you're putting a single template in a single partitioned droplet and that reads as a positive, you are now you're counting the number of positive, you're counting the number of molecules that you started with.
Thank you very much. So our next question is, if I move on quickly, what are some benefits of using ddPCR versus next-gen sequencing other than lower sensitivity?
Good question. So we kind of see ddPCR and next-gen sequencing as complementary technologies. Next-gen sequencing is generally a very broad, if not quite as deep measurement, whereas ddPCR is a very, very, very deep, though often somewhat narrow measurement. We can get much more sensitive. The other benefits are with next-gen sequencing, the process of library generation itself, there's several rounds of amplification. And so amplification adds bias. And once you start adding amplification, even with molecular barcoding, you're still getting relative measurements rather than absolute numbers of copies. Thank
you very much. So our next question is, can you measure BCR ABL?
Yes, we can. Actually, it's an area of hot research. There's a lot of discussion going on right now amongst many labs for translocation testing or BCR-ABL testing, for instance, and bringing BCR-ABL testing and using digital PCR to not just standardize, because there's an IS standard for that, but to really make each of the machines across wherever country you happen to be and testing center you happen to be give the same kind of numbers, so that if you go from one hospital to another hospital and your data goes with you, you can track or monitor your CML and not have your numbers jump horribly between centers.
Excellent. Thank you there, Dr. Shelton. So if we move on now, we've still got a few questions coming in. Our next one is DNA extraction a key step in order to perform ddPCR on a circulating tumor DNA from plasma samples?
I mean, it is. I mean, DNA extraction or any kind of sample extraction is always a critical feature of how well PCR will work of any sort, digital or not. We're not any more sensitive to any particular kinds of extractions. The one caveat to that I would say is that the use of high levels of detergents, if those detergents are left in a sample, like we do have people who are doing crude lysates, for instance, but especially in the plant world, and as well and is that enzyme, the TRAP assay, uses detergents as well. And if you have a too high a level of detergents, similar to cells, the detergents will lyse the droplets. And so there's some kind of, you have to be careful about that, and there's some tests that you run extra to make sure that's not happening to harm your analysis. But beyond that, nothing more unusual than standard genomic practices.
Thank you very much. Our next question is, could it be feasible to apply ddPCR to detect circulating tumour DNA samples? Is that the one I just read? Circulating DNA in plasma samples in a POC device. So can you apply ddPCR to detect it in a POC device?
Extrapolating into the future, maybe. Right now, the current device is not a point-of-care device, really. If we're talking about point-of-care devices similar to, some of the viral or infectious disease tests that are out there, the reading of the droplets, as I said, is like similar to a flow cytometer in cell biology, and similar to that, it's a kind of a bulky machine in that sense.
Thank you. I think we've got time for a few more questions that are coming in now. So our next one is, for the KRAS multiplex assay, how can we validate for the individual assays within the multiplex?
Oh, sorry, yeah. Again, as I mentioned in the data presentation, we have the duplex or the singleplex assays. The individual mutations are wet lab, wet bench validated already and purchasable on the Bio-Rad website, and they will give you the exact same numbers.
Thank you very much. Moving on, a similar question here is, in one of your slides for the KRAS assays on different sample types, the FFPE sample shows intermediate droplets between the blue and black drops. What is this due to?
Yeah, we call it rain, and the intermediate droplets are the intermediate fluorescence amplitudes. It means that the DNA that's being amplified in that particular droplet doesn't have maximum efficiency. Maximum efficiency could be inhibited by either chemical inhibitors. FFPE in particular often has chemical inhibitors that harm the PCR reaction or cause a little bit of a problem. I think the main reason is really the formaldehyde fixation of the DNA causes cross-links. So the cross-linked DNA, you now have duplex DNA, for instance, that doesn't want to separate during the melt. You also have tertiary structures where 4 or 5 strands of DNA are bound to each other, cross-linked to each other, and that makes it harder for the PCR reaction to occur at full efficiency, and that's what causes the intermediate rain.
Thank you very much for that response. So moving on, you mentioned copy number and mutation detection assays for digital PCR. Do you have any gene expression assays available for ddPCR?
Absolutely. So recently released on the website is a I don't even remember how many thousands and thousands of gene expression assays. The nice thing about gene expression assays is that they can be multiplexed, 3 or 4 different targets or references, however you want to do your combination, in one well fairly easily.
Okay, thank you, Dr. Shelton. It appears that that's all of the time that we've got to go through with regards to questions. So if we didn't get round to answering your question today, then your question along with your email address will be forwarded onto our speaker and Bio-Rad, and you'll receive a response in due course. So just before we conclude things, Dr. Shelton, do you have any final thoughts that you wish to leave with the audience?
First off, I'd just like to say thank you for your time, and I hope you will contact a Bio-Rad representative if you have any questions. We are often at many of the cancer conferences as well as viral conferences and the rest, and it's a very easy thing to learn.
So that just leaves me to thank Dr. Shelton for what was a great presentation, and thanks to Bio-Rad for sponsoring our session today. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.ddPCR.com. business-review-webinars.com. Should you like to meet a Bio-Rad technical specialist to discuss how ddPCR can contribute to your research, please indicate this in the post-event survey that will appear on the screen once this broadcast ends. For more information on Droplet Digital PCR and educational resources, please visit www.bio-rad.com/webinar. This URL is also located within the green resource list widget towards the bottom. We look forward to sharing further webinars with you, so please do keep an eye out on our website that was just mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you once again, and I hope you all enjoy the rest of your day.